Hello everyone, good afternoon. Please, all the ones in the back, come in, find your seats. And we would like to start with the second keynote of today. And it's my real pleasure to introduce Professor Simon Eickhoff, who will be giving this lecture. I know Simon for quite a while now. He's one of the colleagues I know the longest. And so it's a real pleasure to introduce him here. Simon studied medicine in Germany and the UK. And then already during his medical studies, he joined the lab of Carl Zillis at the research center in Jülich to do his doctoral thesis work, where he started off with basic neuroanatomy, cortical mapping. And then already during his medical studies and his doctoral work, he became very interested in neuroimaging. And that is probably the part where he contributed the most over the past, we just talked about 16 to 17 years within this organization already. So his contributions vary quite a lot. The thing that probably most of you know is the anatomy toolbox, which Simon already invented during his doctoral thesis and is contributing to it and working on it ever since. And it can be downloaded for free, so a lot of you probably work with it. After finishing his doctoral thesis, Simon stayed in Jülich, became a postdoc in Carl Zillis' lab, and worked on with particularly functional neuroimaging data and with meta-analyses. And with meta-analyses, he gave his second major contribution to the field. He worked on and on with that and is one of the major developers and contributors to the ALE meta-analytic approach. And probably a lot of you already know he was awarded the Young Investigator Award, particularly for this contribution in 2015. He moved on with the functional imaging and combined that also with other parts of imaging analysis, and always interested in connecting the structure and functional relationships in the brain with mapping aspects. And as it moves on, he's now becoming more interested in so-called modern approaches, machine learning, big data analytics, and to apply that to the imaging world. And over the past years, Simon had several positions, first as assistant professor in Aachen University, and then as associate professor in Düsseldorf, where he built his own group, and the group increased in size even more over the years. And which is even more fascinating, that he now started his own lab in Jülich and at the University of Düsseldorf. At the beginning of last year, he became full professor for neuroscience at the University of Düsseldorf and is now heading the Institute of Systems Neuroscience at this university, and at the same time became director of one of the institutes for neuroscience and medicine at the research center in Jülich, which is called Brain and Behavior. So it always gives a promise to what we are looking at, and it is a real pleasure to introduce him here, to welcome him on the stage, and we are very much looking forward to your talk, Simon.
Well, thank you, Svenja, and a big thank you to the organizing committee for inviting me to give this lecture. I guess that in spite of all the different approaches and methods that you in the audience use, we all have a common goal, which is to understand how the human brain works. Now, what that actually means probably is quite different between all of you, but I hope that we can agree on that there's at least two fundamental aspects or themes. One is to elucidate how the immense intersubject variability in structure, function, and connectivity of the brain relates to aspects such as age, gender, genetics, and how it influences, on the other hand, cognitive, socio-affective, or clinical phenotypes. Now, these questions are more and more being answered now, or addressed at least, by the use of big open data sets, machine learning, and high computation power. Now, I said this is just one of the two big themes. The other one, which is really also historically very much at the heart of this organization, is to understand how the brain is organized. And my main push towards this talk will be that I think we should integrate both of them more, because they can both inform and complement each other. And I will start with one of the things that makes the brain so special. In contrast to any other organ in the body, the brain is topographically organized. That is, it is made up of several distinct units and cortical areas. And the key idea is that each of these areas is distinct from its neighbors in terms of its structure, both micro and macrostructure, in terms of its functions, and in terms of its connectivity patterns. Importantly, macroanatomical landmarks, gyri and sulci, are only loosely related to these functional building blocks of the brain. That is, based on gyri and sulci, we can roughly estimate where an area it is, but gyri and sulci do not conform the functional territories. So how do we delineate these? The classical approach that's been around for more than 100 years, and that was also my start into the field, is by histological mapping, either, for example, with receptor architecture or with cell body staining. And in Jülich, this has a tradition for way more than 20 years now. And what we're doing is to map human postmortem brains and then transform them into the MNI reference space and overlay them. So we can get these maps that tell us how likely do we get a particular microstructural area at each location in the MNI space. Now, as Svenja already mentioned, we've provided the SBM Anatomy Toolbox as a tool to do structure-function relationship analyses, and these maps are now also integrated in other softwares such as FSL and AFNI. If you're interested to learn more about the newest developments, that's the integration in the Human Brain Project viewer. You can find the poster number on the bottom right of the slide. As a complementary approach, however, we can also map the brain in vivo using connectivity-based parsilation. The key idea here is that we have a region of interest, as indicated here in the violet color, and then for each individual voxel in that region of interest, we compute the whole brain connectivity profile. From this, we then can compute the connectivity matrix for the number of seed voxels times number of voxels in the rest of the brain, and a cross-correlation matrix telling us how similar are each pair of seed voxels in terms of their connectivity pattern. Using unsupervised learning, clustering, we can then group the different voxels such that voxels with a similar connectivity profile are in one cluster, whereas the connectivity profiles between the clusters are as different as possible. If we map that back to the brain, we get something that is very similar to what we've just seen from the cytoarchitecture. And that is not a coincidence, because in the end, what we're doing with either cytoarchitecture or connectivity-based parsilation or any other method is really we're looking at one particular feature of brain organization, and then we either try to identify borders or groups, but in any way we try to delineate territories based on that particular feature. Now then the big question is, what are these regions doing, right? Well, in some cases it's very easy. What's the function of Broca's area? Right area, left area 44, 45. I think everybody of you knows what Broca's area is doing. It's visual search primarily. And some motor imagery. Well, there's also action observation, mental algebra, spatial mapping is a big part of it. Maybe some of you even thought about language. Now, what's the point here? The point here is that if you search the literature, given all the variance that there is there, you can interpret any location of the brain in terms of any function you want. You will always find at least one paper, I guarantee you, that exactly maps this function to that region. Is that where we want to go? Well, most likely not. So wouldn't it be nice if instead of searching the literature for something that fits your ideas, you could just objectively say, well, what are the functions probing that activate this particular part of the brain? And one way to answer that is to resort to activation databases. There are two of them, two large ones, BrainMap and NeuroSynth. I will focus here on BrainMap. So this is a database where you have thousands and thousands of experiments, and for each experiment you have where did it activate and what did it do. And then you can basically statistically answer the question that we, in a little bit of a joke fashion, answered subjectively. You say, well, this is my region of interest. Now you extract all the studies in the database, you filter the database by that location, and then do an inference on the particular functional metadata. And I'll show you a couple of examples now throughout the talk. Whenever you see the little brain map logo, that's when I use functional attribution that's actually based on statistics rather than guesswork. Now let me show you an overview on how brain mapping can ideally work. This is an example from the frontal pole of the human brain, which we mapped both using histological analysis in Katrin Amund's lab and using connectivity-based parsilation. And quite nicely, we see a convergent distinction into a frontopolar part, that's the lateral part, FP1 in blue on the slides, and a more mesial part in red. If we then look into the brain map database, we see that they are not only different in terms of their structure, but also in terms of their function. The blue lateral part is involved in cognitive functions. The red, the medial part, is involved in emotion and social cognition. And indeed, they also do interact with very different networks. So for example, the cognitive area interacts with the parietal cortex, the red one, the mesial one, with the amygdala. Now we can also go now into the clinical domain and ask, does that have a relevance? Here we studied about 80 patients with major depression compared to healthy controls. And what we saw is that only specifically the medial area, the red one that was involved in emotion processing and connected to the amygdala, only this area was affected by major depression, and that this affection also correlated with disease severity and duration. Now what's actually quite interesting, now we took the reverse approach and said, well, can we classify patients from controls using a searchlight analysis? So we just went through the entire frontal pole and basically with a regional searchlight said, can we classify patients from controls? This was completely agnostic to the histological and connectivity-based mapping, and yet again you see in yellow there's a high accuracy, in red there's a low accuracy, and again you get the distinction between the medial and the lateral parts of the frontal pole. Final part of the puzzle, Sebastian Bludau now has a paper just published in Brain Structure and Function, where we used these maps to query the Allen Brain Atlas, and we could show that these two areas also differ with respect to gene expression, among other genes also for the MAO gene. So actually there is a whole connection. Now this is the best case scenario. This is how we would envision the brain. Unfortunately, it seems that, well, first of all, it seems that this thing isn't working. Okay, that this is not always absolutely the case. Here you see an example from our current work that is also at display here on the conference on the hippocampus. And here for the comparison, you see the cytoarchitecture of the hippocampus. And what you can see is that whereas the cytoarchitecture is really organized in a medial to lateral fashion, if we look at a connectivity-based parsilation, we also see differences between the anterior and the posterior part, which actually fits quite well with data from non-human primates and other animals, showing with tracing studies differential connectivity of the anterior and the posterior part of the hippocampus. So what this suggests, I wouldn't say it definitely indicates, we definitely have way more work to do on that. What it may suggest is that, quite often at least, all features change at the same border. But in other cases, you can have somewhat like a grid-like arrangement. So for example, in the hippocampus, the cytoarchitecture has the one axis, and you have an almost perpendicular axis on connectivity, and together they form functional territories. It's a hypothesis. We're going to test that. But following the same line of ideas, we also had a look at the chordate, and again you can see different parsilations based on resting state, diffusion imaging, and structural covariance. And what we did here is to start exactly the idea that I just mentioned, and now apply a consensus clustering, where we say, well, if we take all the information together, what kind of map will we end up with? And this is what you see over here. So where does it lead us in terms of topographic organization of the brain? Ideally, what we would want is something like a universal map that optimally captures variance in all features. Why do I call that a utopia? Well, the problem is we don't even know how many features there are. We can look at so many different things in the brain. And so from that perspective, I'm not quite sure we will ever get to this perfect universal map for which we would probably need to look at dozens and hundreds of different features, histological, neurochemical, trajectories in aging, in development, and so on and so on. If we ever come up with one map that captures all of that perfectly, again, that's why it's a utopia. So what we can do more practically, however, is to work with unimodal maps and then do cross-modal evaluation. Now, I've already shown you an example of that with the frontal pole. There was a map that was originally based on cytoarchitecture, and then we looked at different other things, connectivity, function, depression, gene expression, and in fact, indeed, the cytoarchitectonic border also coincided with differences in these things. Another project where we followed a similar approach is the work on the Brainatome Atlas led by Tianzi Jiang, which is a diffusion tractography-based parsilation. And then for each of these different areas, we also looked at their functional connectivity profiles, at their functional response characteristics, and so on. So this is a step towards that idea of having a map in one modality and applying it to a series of other modalities. Another example for a unimodal atlas, and that's what I'm going to talk about a bit more in the next couple of slides, is work led by Thomas Geo, and this is the, I think now called Schäfer parsilation. This is based on resting state. Now, we would assume that a resting state-based parsilation is an optimal data compression for resting state, right? Because it's intended to have homogeneous resting state patterns within and different resting state patterns across parcels. So let's use that to look a bit more into resting state and come back to what I said at the very beginning, look at inter-individual variability. So as many of you are probably familiar with, from these parcelations you can extract these kind of connectivity matrix. Now, you will see a lot of work here, and we and others have done a lot of work on using this matrix as a whole to predict phenotypes. I think there are at least 2, maybe 3 or more fundamental problems, issues, drawbacks of that. One is dimensionality. This thing has way more features than we would reasonably even dream about having subjects. And on the other hand, if you then get a good prediction, a good classification that is based on a distributed pattern across the entire connectome, interpretation becomes very difficult. And remember, in one of the earlier slides, I had these bidirectional arrows. We want to go from brain organization to prediction. That would be basically this step. But we also want to feed back. We want to learn something about brain organization from predictive analysis. So what we did here is to actually go in a row-wise fashion. So in the poster indicated here, we just took for each parcel the connectivity profile with the rest of the brain, and then asked, can we predict the sex of a new unseen subject by these region-wise connectivity profiles? And that was across 2 different samples, so a fairly conservative analysis. And what you can see is that there is quite some interesting regional variation in the predictive power. It's in particular regions of the cingulate cortex and of the left hemisphere, in particular frontotemporal, whose connectivity profiles allow you to predict a new subject, whether it's a male or a female. Now, if we then look into BrainMap to see what are these regions doing, we actually found a very nice convergence again. These are regions involved in language, emotion and reward processing, and cognitive control. Encouraged by that result, we also looked into a clinical domain, and in particular into schizophrenia. So I have to go a little bit back. We have a large sample of schizophrenia patients, and as you can imagine, and probably most of you know, schizophrenia pathophysiology is unclear, and psychopathology is chaotic. every patient kind of has its own mixture of symptoms. Now, we first of all wanted to sort that chaos a little bit, and the way we did that was by applying non-negative matrix factorization to psychopathological data. So what you see here, and I'm sorry if you can't read that in the back, and I will explain it to you, is a cross-validated low-rank description of psychopathology in about 2,000 subjects that shows that you can represent psychopathology with 4 major factors. There is a cognitive, an affective, a psychotic, and a negative factor. Next, we went on and then clustered patients into groups to see where can we, in this now much more manageable and also somewhat denoised space, find subgroups of patients. And indeed, we could find evidence for 2 core subtypes. It was basically a more positive, a more psychotic type, and a more negative, a more blunted type. Now we went back to the imaging data and did exactly the same analysis that I just showed you. And like for the sex classification, we again found very distinct patterns. So there were some areas where the connectivity profile was very well discriminative of the 2 subgroups, the psychotic and the negative one, whereas most of the other areas are not. And now you may wonder, why are so many here actually in gray? Well, all the areas in gray are actually below what we can get in terms of accuracy when we use the entire connectome. In reverse, that means if we don't use all the information, but if we use these regional profiles, there's a lot of regions that do better than whole connectome-based prediction. That may be somewhat counterintuitive, because if you do a whole connectome, you can find the relevant connections in there. But remember what I said about the number of features relative to the number of examples. Basically, what we can conceptualize this kind of approach as is a biologically informed feature reduction. Now we are reducing the problem to a lower number of features, and we are actually better in predicting, in this case, clinical subtype. And if we then look again, what are the top predicting regions doing? It's actually very interesting. So this one here on the temporal-parietal junction is involved in social cognition, theory of mind and face perception. This one here on the subgeneral ACC involved in reward, reasoning, and mood induction. So it actually also can map quite well onto the sort of negative affective versus the more positive psychotic subtype. Encouraged, we moved on, and remember what I said about cross-modal evaluation. Now the question is, this is a resting state parcellation. It was intended and set up to represent resting state optimally. But does it also represent structural imaging data? It would be nice if it did, right? Because then we could again use the same atlas, now query a different modality. We addressed that question by using a predictive framework, in particular the so-called brain age estimation. The idea is quite simple. You take a structural description and you basically want to predict the age of an unseen subject. We just use a repeated cross-validation to do that. Now, as the sort of comparison, as the gold standard, we use the factorization of the VBM data. So what you see on the left should optimally compress the VBM data. What you see on the right is actually meant to optimally compress the resting state data. Now, what do we get in terms of age prediction accuracy? For the VBM compression, we get an accuracy of about 6 years. And what we get when we use the resting state atlas, again, it's basically identical. Similar, when we look at data from the ADNI sample, normal subjects, mild cognitive impairment, and Alzheimer's, we see an advanced brain age in dementia when we use the VBM compression, and we see basically the identical advanced aging in Alzheimer and MCI when we use the resting state data. So this seems to indicate that indeed there is enough transfer of data from one modality, of a parcellation from one modality, to also look at other modalities. So we implied that, somehow the thing is a bit tricky. Can you tell me where to point that to? Okay. Well, let's see how that works in the future. So here we applied that to the analysis of heritability. So as probably many of you know who are in the field of imaging genetics, usually what you investigate is free surfer atlas, desiccant atlas regions. Now they're based on macroanatomy, and what I said earlier probably makes you wonder, is that the optimal thing to do? Because macroanatomy is not related to any biological territories in the brain. So now we used, as an example, the heritability of cortical thickness and surface area, now based on the parcel-wise, on the resting state parcellation. What we can see here is that indeed we get a very sharp profile on very differentiated heritability. Now what's interesting now, because we are in the same space as we did for the gender classification, as for the schizophrenia, now we can integrate all of this information. At some point we're going to build up a big per parcel database that gives us a lot of information. I think cross-referencing all of these information, that's hopefully going to advance our understanding of what is this part of the brain really for. Extending that work, we also then looked at co-heritability between brain and a behavioral phenotype. In that case, there was personality traits from the neo-FFI, and we saw, for example, a very specific co-heritability between extraversion, the trait of extraversion, and cortical thickness in only two parts of the brain, marked here in yellow on top. In particular, the strongest hit was the temporoparietal junction, and this area, very nicely, in the brain map database, was associated with social cognition. To sum up that first part of the talk, topographic specialization is one of the defining principles of brain organization. It's present, it's probably present, I would say it's present, in all neurobiological feature, microscale, histology, molecular, all the way up to system level. It's not reflected well by macroanatomy, but it seems to have specific relationships to both behavioral and clinical phenotypes. Now, the classical concept is that areas are distinct from each other in structure, function, and connectivity all at the same time. And this is indeed reaffirmed by a lot of studies. And what I think is that in some discrepancies that we see, these are actually the most interesting regions because they tell us really something about brain organization now. So I would argue and hope I have convinced one or the other of you to really move towards this field, combining cartography and predictive analysis. So this is relating big data analysis, machine learning, to brain organization on the map side. But we also have the other side, the side of distributed networks, task activations. There's a huge literature, and people also on these conferences are using many different tasks to probe brain responses to various stimuli, contextual demands, and so on. The question is, how much can we actually trust that? Well, the answer is even a bit more negative than I hoped it would be. Well, it was even more negative than I feared it would be, to be honest. As you see in this analysis over here, now that's a whole brain BBN analysis, and we just had a look at a quite big data set, and then basically the split half replicability. And what you see in these turret diagrams there is the distribution of overlap. So how many voxels overlap? On the left, you see an analysis of age effects. And you see a lot of voxels that are sort of on the ceiling. So that's the red ones on the image. So in a lot of voxels, we see a good replicability in the split half analysis. Now, if you look at the right, you see the findings of perceptual IQ. And I'm sorry, but I think you cannot read the numbers there. So the right number is n equals about 140. So split half is 70 subjects each. Top replicability is about 15%. The leftmost bar in the green one on the right plot is 330 subjects. So it's more than 150 per subgroup. Top replicable voxel is about 70%, if I'm generous. So we do have maybe a little bit bigger problem of replication than we previously thought. And this is actually worse for fMRI. Why? Because we all have the additional factors of heterogeneous paradigms, analytical flexibility, small samples, and quite a bit also publication bias in there. Now, coming back to the question, can we trust a single fMRI result? I would say probably not too much. I mean, I'm happy for everybody, my students included, if they have nice fMRI results, but I wouldn't put all that much trust in a single study. The thing is, however, that this pointer really doesn't work well. We have a noisy, but also a really big literature. There are way more than 30,000 fMRI studies by now. Well, if we can't trust a single study all that much, maybe across studies, something that is robust, that comes up over and over again. That's something we should probably be able to trust a bit more. And this is the field of neuroimaging meta-analyses. The two fundamental aspects, you have either image-based meta-analysis or coordinate-based. And here I'm really only talking about coordinate-based meta-analysis. So it's only based on the XYZ coordinates that are reported in the papers. That is an extremely sparse representation, but it has the advantage that we can get back all the way to the initial studies in the early 90s, because we don't rely on the authors actively sharing and cooperating, because the coordinates are in the papers. So how does coordinate-based meta-analysis work? I'll show you an example here. These are the activation foci reported in about 200 working memory studies, and you see them scattered quite a bit around the brain. Next thing that we do is to compute a convergence map, the activation likelihood estimate, after accommodating the spatial uncertainty of the foci. And then we can actually draw statistical inference that based on a permutation-based null of random spatial association between studies. So we get from the quite heterogeneous scattershot picture on the top to the bottom, which is now what I would call the robust location of working memory areas. These are areas across many studies that over and over get activated by working memory studies. The field of meta-analysis has exploded, and it has been particularly successful in the clinical domain, where, among other things, we found convergent correlates for many disorders, and as you see on the left here, I don't click back because I don't know whether I can move forward again. As you see on the left, we even find convergent neural substrates across many different psychiatric disorders. So there seems to be something like a core neurobiological phenotype of psychiatric disorders, including schizophrenia, bipolar disorder, depression, thanks a lot, and so on. Now, this has almost put the meta-analysis field in a very problematic position. Why? Because at some point, we were facing the criticism, you always find something. It's trivial. And so I was very lucky, very happy, that we had two major null results. On the top right, you see the convergent neural substrate of depression in fMRI studies. And now if it looks odd, well, there is nothing. So if you look across more than 100 fMRI, task-based fMRI studies in depression, no convergence. The lower is the convergent neural substrate of the COMP gene influence on working memory activations. And again, there's nothing. And what I thought was extremely encouraging is that actually quite good psychiatric journals were open to also publish such null results, because I think for the meta-analysis field, this was critical to show that we're not sort of automatically getting some convergence somewhere all the time. But even with well-powered meta-analysis, you can actually get null results, and I think they tell you something extremely important. Apart from these kind of inferences, the remainder, the last 15 minutes, I will focus on what I think is an exciting new perspective now. It's to use meta-analyses as priors for primary data analysis. So the key idea here is that you have a big and heterogeneous literature, then you do the meta-analysis and get to the robust neural correlates of a particular function, let's say the robust neural correlates of working memory. And they can use the areas you find here as nodes and in individual subject then extract functional connectivity within this network. And what this basically means is that instead of looking at the whole connectome, or instead of looking at the whole brain connectivity pattern of an individual region, we're now looking specifically at the connectivity pattern in a robustly a priori defined network. And as a proof of concept, we actually looked at a whole array of different networks. So that's anything from emotion processing, the theory of mind, empathy, reward, memory, cognitive action control, and so on. And what we looked at is how well does each of these networks classify Parkinson patients from healthy controls, matched healthy controls, and how well does each network classify schizophrenia patients from healthy controls. Now, you may wonder why. What exactly is the rationale behind that? The rationale is that we can use that basically to probe each network. How much information about Parkinson is in that network, how much information on Parkinson is in that network, in that, and so on, and conversely, how much information in schizophrenia is in each of these networks. And if you do the analysis, you can actually see very nicely that there's a clear differentiation between networks. So in some way, we're using this network-based diagnosis to reversely diagnose our networks. So again, this goes into this sort of bidirectional loop between understanding brain organization and predictive analysis. In particular, what we see here is that it's the memory, autobiographic memory, and semantic memory, as well as the motor network that is best predictive for Parkinson's, and it's the emotion processing, empathy, and cognitive control networks that are mostly disturbed in schizophrenia. And so this actually fits also a lot of the behavioral characteristics quite nicely. What was a bit of a sort of hold onto our enthusiasms to rush to the clinicians and have them implement that is this finding. Any network is better at classifying old from young subjects than it was in classifying any disorder from healthy controls throughout. So I think we are still, in spite of all the work that's going on there, we're still underestimating the effect of covariates, in particular age, on prediction work. Let me show you another example, very much along the same way, but now we're not doing a binary classification. We're not saying how much does this network differentiate patients from controls, but now we're trying to predict individual phenotypes from network-specific activity, connectivity. Here are two example networks: cognitive action control, based on a meta-analysis, and socio-affective default mode, again, based on a meta-analysis. And we choose these because they are basically sort of the opposite poles of what the brain can be in. Now, how well does the connectivity pattern in this network, the individual subject-specific connectivity pattern in this a priori defined network, allows us to predict something like working memory, spatial working memory, the Corsi block tapping test. And what you see here is a very good prediction accuracy for the cognitive control network, but you see a complete absence of predictive power in the socio-affective network. And in case you wonder what these large error bars are, this is variance in the prediction based on the initial 10-fold split. So we actually replicated the predictive analysis 250 times, and these are the error bars just due to the initial folding of the data. We went on, tested another cognitive function, this time dual task costs, same network, different phenotype, but obviously related. And again, we saw that the cognitive network fairly well, depends on the point of view, allowed us to predict cognitive performance on the individual level, whereas the socio-affective network did not allow that. Next on, we saw, well, what happens if we split the subjects? Because I didn't tell you that. This group was actually made up of two subgroups, a younger and an older one. And if we look at the same behavioral phenotype, dual task costs in the young and old subject, and this time only in the cognitive control network, we see it actually predicts performance quite well in both of these groups. In fact, if you restrict the analysis to either the young or the old, the correlation between true and predicted actually becomes even a bit better. However, if we look at another phenotype, and this time it's a composite score of working memory, same network, we see that it is only predictive and quite nicely predictive in the older subjects, not in the younger. So what I think is that we're just starting to open a really big bottle here, because we do have a lot of different networks. We have now quite a bit of evidence that there is differential information on clinical and behavioral phenotypes across networks. We see that, let's say, it passes the eye test. It's the cognitive networks that allow to predict cognitive function, but not the social affective. It's the motor network that has a lot of information about Parkinson. The emotional networks have more information about schizophrenia. And we are starting to be onto something, but I think this data, which is actually presented here also in an oral presentation later this week, shows that we have to be a bit more careful because there's a lot of nonlinearities in all of this data. Last but not least, I think the critical question, is that actually better than whole connectome prediction? Remember I told you earlier when I talked about the parsilation-based work that if you go by parcel-wise connectivity profiles, we outperform whole connectome performance based on the idea that we optimize the number of examples, subjects, relative to the number of features. So the same logic, I guess, should hold here as well. If we're restricting ourselves to a smaller network, then we have fewer features, so a better sample to feature ratio. However, this is obviously only helpful if the features within one network are actually predictive. So what I'm showing you here is data that was just published on the neo-FFI personality scores. And I, for the interest of time, just show one here, and that's the personality dimension of openness. Now, what you see here, the circles in the middle, that's the classification performance using whole connectome classification. So what you see quite nicely is that a series of networks substantially outperforms, these are the individual networks, it's a couple of networks substantially outperform whole connectome prediction. So the most striking example here, the big green, what is it called, wedge on the top left, that's the reward network. And so if you look at the connectivity pattern only in that network, the accuracy is almost 3 times as high, the correlation between true and predictive scores, as compared to using the entire connectome. And that's obviously replicated 250 times, so we are sure we're not getting into some effects of folding. What's quite interesting, what we also see in that data, so I already showed the old-young problem, now we see here that we also have a quite strong sexual dimorphism. So this particular network actually allows to predict personality scores, the openness score, significantly in the females, that's in green, but there was no predictive power at all in male subjects in red. Now, did we just get lucky on the females? Well, we wanted to find out. So this was an analysis based on 210 unrelated women from the HCP dataset, and then we replicated that in another dataset of 150. Different, again, unrelated women, and we find exactly the same pattern. So overall, what I would like to propose is to integrate between network mapping, not brain parcellation, but network mapping, as we can do by meta-analysis, to get robust networks, to get the robust nodes of task-specific networks. I think that's the key here. We later analyzed task-free data, resting state data. So what we can do here now is leverage the information from thousands and thousands of task-based neuroimaging studies, find those regions that are robustly engaged in a particular task, and then extract task-free data, in this case resting state data, within that network, use predictive analysis, and then learn something both about inter-subject variability, but also learn something about the network. So I hope it became clear that throughout the talk that the underlying topic is we need to bridge these sort of big data, machine learning, predictive analysis, and the detailed look at brain organization. And in keeping with something that was a big topic here in Singapore last week, I would suggest that we need to more reconcile the precise deep neuroanatomy with the big data science approaches, because the brain is not just a feature matrix. It's actually something that's interesting in itself. Brain maps can provide a biological data compression that is actually meaningful and that transfers also across modalities. Understanding pathophysiology needs biology. If we just have a predictive model that is based somewhere loosely on the entire brain, that's not going to advance our understanding. It can be useful, but it's not helps us to understand brain organization or pathology. But predictive power can give insight into brain organization. This is what I showed when I showed you the gender analysis, the schizophrenia analysis. Finding out which passwords have a predictive power informs you about the password. Atlases allow the multimodal integration of data. Meta-analysis then yield robust task networks that we can use as prior knowledge to boost accuracy and interpretability of task-free predictive analysis. And so I will conclude with, in order to understand how the brain works, how it's different between subjects and how it's organized, we need to push this integration between understanding neuroanatomical details, functional neuroanatomy, and big data predictions. And with that, I would like to close by thanking everybody who has been involved in the various projects, in particular, obviously, Katrin Amundsen and Carl Zilles, who have been involved the longest, all the collaborators, all the people that are listed here on the right that actually perform the individual studies, as well as the entire team. Thank you.